Hi, everyone. Welcome to our webinar. My name is Kasia Proctor, and I'm the Director of Marketing Communications here at Biolog. I will be your moderator today. Our webinar today is titled Microbial Mixology, Comparing Communities Across a Range of Ecosystems with EcoPlates. I have a few housekeeping items to cover. The recording is going to be available to everyone who registers. And if you have any questions, please submit them through the Q&A chat feature. Now let me introduce our speaker. Luis Cantu is part of our Biolog family here. He is a field application scientist. He is in direct contact with many of our customers, answering their questions and supporting their research. And in his spare time, he likes to hike around the Bay Area or play guitar with his friends. He earned his PhD in microbiology from UC Berkeley. Welcome, Luis. Take it away.
Thanks for the introduction, Kasia. And thank you for joining this webinar. Today we're going to talk about how EcoPlates are used to compare microbial communities. They are really powerful tools for measuring microbial activity and to understand how these communities respond across different environmental conditions. Let's get started. So microbes are everywhere. For example, in the vicinity of this plant, we have many different microbial communities. These communities are highly dynamic systems where microorganisms are constantly interacting with each other and with their environment. For example, in plant-associated microbiomes, microbes often interact with high-order organisms like insects, and they provide protective benefits like antibiotic production. But microbes also play critical roles in nutrient cycling, helping make nitrogen, phosphorus, and other nutrients available to plants. They also contribute to carbon cycling, including processes like carbon fixation and decomposition. They even help break down complex organic compounds, such as phenols, polymers, and other contaminants. This process is important for biodegradation and ecosystem recovery, especially in disturbed or polluted environments. And like I said, these microorganisms are everywhere. They live in soils, oceans, plants, animals, and even in extreme environments like deserts, caves, and deep sea vents. And microbiomes change across environmental gradients, disturbances, and host-associated shifts. When we study microbial communities, we're not just looking at which organisms are present. We're trying to understand what functions they perform and how these functions change across environments. And over the last decade, sequencing technologies have transformed microbial ecology. Sequencing allows us to answer the question, who is in the community? For example, this recent paper explores how Clostridium butyricum improves pig health based on in vivo and in vitro studies. With 16S sequencing, we can see how the microbial community is changing when we add a new member of the community. And we have many ways to show these population shifts and restructuring at different levels. For example, at the phylum level, there might not be very evident changes, but at the genus level, you start to see major shifts. And at the species level, we see very, very big changes. But that leads to an equally important question. What can the community actually do? How does the functional potential shift over time or conditions, right? What does it mean if there are changes in the community members to the overall metabolic activity? Are specific members responsible for key functions? To answer these questions, we need functional measurements. And this is where functional profiling tools like EcoPlates become extremely valuable. EcoPlates measure metabolic activity of microbial communities across a panel of carbon substrates. So they contain 31 environmentally relevant carbon sources, each replicated in 3, plus a control well. When microbes metabolize a substrate, respiration reduces a tetrasodium dye, producing a color change. And this allows us to measure community-level metabolic activity using absorbance. Now, the substrates represent different functional groups. So these are the 31 substrates, and they're organized by different functional guilds, including carbohydrates, amines, phenols, polymers, amino acids, carboxylic acids. And one strength of the EcoPlates is their flexibility. They work across different sample types, like soil samples, plant-associated microbiomes, aquatic samples, and animal-associated microbiomes. And our Odin platform allows researchers to compare microbial function diversity across very different conditions in a high throughput manner. EcoPlates experiments often benefits from our Odin instruments because it's an automated phenotyping platform. These systems allow researchers to monitor growth curves in real time, measure respiration kinetics, run multiple plates simultaneously, track metabolic responses under different conditions, and more importantly, they allow us to capture kinetic data, which provides deeper insights than just single point measurements. Let's discuss some recent publications across different applications. I have 4 case studies here that include ecoplates being used in extreme environments, in decomposition systems, in sustainable agriculture, and in restoration ecology. Ecoplates have been used to study microbial communities in extreme environments such as ice caves. And this is a very extreme cold environment, very hard to reach. You can imagine there are not many plants inside, but we can still find microbial communities thriving in there. And in this study, functional guild grouping revealed that microbes efficiently metabolize organic acids and amino acids, but they show lower utilization for carbohydrates and organic compounds. This suggested that the microbial community had adapted to organic nitrogen availability in the cave ecosystem. This is because there's very little decomposing material around it, with no plants or no animals nearby. In this second example, ecoplates were used to study microbial activity and decomposition systems. This study is a seasonal dynamics experiment where researchers studied how the timing of cadaver deposition influences biogeochemical changes. So, in other words, when is the cadaver placed on a soil is going to matter to its decomposition. The study revealed that microbial communities near this cadaver decomposition island showed a strong utilization for carbohydrate substrates. So remember in the previous example, there was no mammals nearby decomposing and they switched to a different substrate, right? But now they're switching to carbohydrate substrate. And this reflects the presence of labile organic compounds released during the decomposition process. Also, the timing of the microbial activity differs significantly between warm and cold seasonal conditions. Basically, decomposition occurs much later in the cold weather than in the warm. And it seems to be related to the community is better adapted to warmer weather so that their microbial activity is higher during warm conditions. And ecoplates are also widely used in agricultural research. For example, researchers studying Mediterranean olive farms used ecoplates to measure functional diversity of soil bacterial communities. And careful sample preparation was essential in this paper, including soil dispersion and normalization of the bacterial density before inoculation. And this technical note does a great job at documenting their whole workflow in very great detail. The authors of this paper actually developed a metric called average well color development, and it is used to measure overall metabolic activity of a microbial community in soil. And we have incorporated this metric in our built-in analysis tool of our Odin instrument, and we'll talk about it more in detail in later slides. This is a very nice technical note that I always recommend our customers so that they can start designing their own experiments. And so in the study, like I said, they use CFUs or plate counts to estimate a bacterial density, and they documented how that is done. They prepare soil suspensions using this buffer that seems to be working great for soils. And then they inoculated EcoPlates using this standardized method so that you can get results across different conditions, different soils. Following these steps allows reliable measurement of soil microbial diversity and their function. And this work served as a foundation for agricultural research to continue monitoring soil with our EcoPlates. Their methods are very detailed, and I really like this paper for that. And next, we have my personal favorite. This is a restoration ecology paper. And in here, the authors used EcoPlates to study the microbial response to environmental stressors like nanoplastics and antibiotics. And so it turns out that nanoplastics are these small plastic particles that are being released by, our current usage of plastics, and they can actually influence how nutrients are available to microbes. And similarly, antibiotics are commonly found in aquatic environments, and there's been many studies before that describe how antibiotics actually restructure um, communities. So in this study, they use 16S. So this figure here is of 16S. And they use 16S to measure the overall structure of the community and who is there. And they use all these different conditions of different um, nanoplastic and antibiotic exposure, either in short term or in long term. And they can see that there's this shift in the structure under the stress, right? And so antibiotic exposure significantly alters the bacterial composition. Some members increase and some members decrease. And so we can see with 16S who is there, who's changing, but it turns out EcoPlates revealed that nanoplastics alone can cause the moderate shifts in functional activity. So you don't, as early as just adding nanoplastics without antibiotics, you already start to see shifts in the community. And another key insight here is that the community response is actually evolving over time. So early responses may differ substantially from later ones. And this is in line with what we've seen in EcoPlates, right? So this reinforces the importance of actually collecting kinetic measurements and time series data for your assays. Now, when working with complex microbial communities, sample preparation is critical, right? So let's discuss some more details around this. And so here's an example of a typical workflow for soil samples. But you can use this three-step approach for other samples, including aquatic sources or like extraction of tissue samples, etc. And what we're gonna do here with the soil is we're gonna start by preparing a soil suspension with your preferred buffer. I always recommend you start with a buffer and don't just use water. There's some osmotic effects that you can avoid by using a buffer, and so it is very important that you start with a buffer. Dispersing then your microbes from the soil particulates and removing the debris. So this is the first step that we call here soil extraction. The next step, we're going to do standardizing your microbial density. And you can do this in many different ways, but in this diagram, we're using CFU or plate counting, and this is similar to what we saw in that agricultural research paper. And so we're gonna plate these dilutions, and we're gonna pick one based on the target density that we are trying to achieve so that we can compare across different plates. We're gonna pick that density and we're gonna load that dilution onto plates. And the goal here is to create a consistent inoculum that reflects the community while minimizing experimental noise, right? So it is important to standardize it. Lastly, we're going to incubate and measure OD using our Odin platform, which allows us to incubate and measure up to 50 plates at a time and analyze your results with the built-in analysis workflow. Now, I have a short video here that demonstrates that process, and I'm going to hit play and walk you through it. So first, I'm going to weigh 5 grams of a soil sample, and I'm going to resuspend that in 40 mLs of saline solution. Again, I'm using saline solution or a buffer to avoid any osmotic effects. After about 5 minutes, the debris is going to settle down and I am going to remove the supernatant to perform serial dilutions. And over here, I'm aiming for a cell density of about 85% transmittance. So with 3 serial dilutions is enough. You might have to perform more or less, depending on your soil. Now I'm using a turbidimeter, but there's many other methods for you to normalize your cell density. This is just blanking the turbidimeter so that I get an accurate measurements. I take my dilution and I measure it and I record that density. Now I'm going to transfer that liquid onto a tray and it's time to dispense 100 microliters per well using a multi-channel pipetter. Now when I do this, I try to avoid shaking the plate or mixing the contents of the plate so that I don't create any bubbles. And that is it. At this point, I put the lid back on and it's time to incubate and measure with the Odin. Now typically you want to do this for at least 48 hours, but you can do this up to 96 hours. And we'll wait for results. Now here are 4 considerations you must always take to maximize reproducibility. So if I was to give you 4 advices on how to set up EcoPlates, these would be it. So first, always ensure you're using aseptic techniques, right? Clean collection tools, sterile containers, clean workspaces, use gloves to protect your sample from contamination, to avoid cross-contamination, right? So second, always adjust the density of your samples. I already said that before, but you have to find a way to adjust the density of your samples. You can use a turbidimeter. This is what we recommend, but you could also use optical density or even colony counts. But you always got to find a target inoculum. Next, avoid over-inoculation, right? So if you're using CFU, count your plates at a countable rate, range. Use replicate plates when you're counting CFUs, if that's the method. Only true discrete colonies, and maybe you want to run a small trials to ensure that whatever density you choose is the density that is going to work for your soil. Number 4, use fresh samples. So you want to avoid storing your samples in the freezer for a really long time. You can use transport storage media. So if you're gonna collect soil, you can put them in bags and special containers. You can refrigerate your samples if you're shipping them or in transit, but avoid freezing and thawing. And this is important because freezing actually reduces the microbial diversity. So you still get a readout of your soil, but you might introduce some variability by doing these cycles of freezing and thawing. Now, there are some special scenarios that might need some adjustments to your workflow, right? So, for example, in low biomass samples, You might need longer incubation times or load your extracts directly onto EcoPlates and avoid the dilution. And then you can measure CFUs or something like that and adjust the density as part of your data analysis later. The opposite is true for high organic backgrounds. You might consider doing higher dilutions or more biological replicates. And this is because these high organic backgrounds contain higher numbers of heterotrophic bacteria, so you get a lot more biological noise. And lastly, over here, the chemical properties of the environment surrounding your community are also very important. And you can consider adding additive solutions to stimulate the growth of your community and ensure that what you're measuring is as close as possible to the collection of the sample that you perform, right? So, for example, you might adjust the pH of your buffer to match the pH of your sample, or you might adjust the salinity, for example, if you're collecting um, from aquatic sources, from marine aquatic sources, or from statuaries, etc. Now, experimental design is important when comparing microbial communities, so we can discuss some common types of experiments that we have seen. And so here I have illustrations that relate to soil experiments, but these apply to any other type of sample sources like aquatic sources or tissue extractions, microbiota, etc. And so some common designs involve comparing communities in a treatment versus control condition. So we saw the example of nanoplastics and antibiotics. We can also do before and after a disturbance. So for example, here I have the case of a control fire or a wildfire where we had data from before, and then this control fire happened, or it's a wildfire, and now you can compare both of them. We can also do seasonal dynamics where you just sample the same soil across different seasons. We can have spatial gradients where you're sampling across different altitudes, for example, here, or it could be a distance to a water source or a distance to a tree, for example. And so that is a common experimental design. And lastly, you can do a successional trajectory where you're studying an ecosystem, for example, soil, and you know there's like a repeatable successional trajectory of the species around this ecosystem, but you also want to measure the metabolic activity and the communities that are there. So these are very common um, comparison types that we see. And one important aspect when designing EcoPlate experiment is replication and have a replication strategy. So EcoPlates already contain a triplicate well for each substrate. And because of this built-in redundancy, it's often better to prioritize biological replicates rather than technical replicates. And so biological replicates capture natural variability between microbial communities, and that is typically the question that you're trying to study. So in our Odin platform, the three replicates for each substrate are automatically aggregated during analysis. And this helps reduce noise and generates a more reliable kinetic curve with its calculated parameters. As a result, you can just focus on the community level patterns rather than individual well variability, right? So this is a very nice feature of the EcoPlates. And kinetic measurements are also another important aspect when you're designing design, right? So why are they important? Well, they capture dynamics of substrate utilization rather than just a single endpoint. These time course measurements also allow you to capture the dynamics of the substrate utilization across time. So you can do both time series with kinetics, and you can identify the optimal read window by doing kinetics. So that way you know when you read the plate, it was already at the maximum uh, height of that curve, or if you need to change the time to read those plates, right? This is important, particularly when you're comparing microbial communities from very different environments, right? So looking at kinetic data provides much more information than simply asking whether a substrate was used or not. And that is because, for example, two strains here might metabolize the same substrate, but they differ in their lag time, right? Or in their growth rate. And so the maximum activity is probably the same, but you're missing out, physiological differences that are invisible to endpoint measurements. So kinetic data is definitely very important here. Now, once your experiment is complete, the next step is converting these plate readings into interpretable ecological metrics, right? So this is my recommendation. Always think about the ecology of these samples, right? And so some of the most commonly used metrics include average well color development, and this is the metric that we discussed earlier that was developed for soils, but it measures the overall metabolic activity across the wells. And so it's a really nice metric. We also have Shannon Diversity Index, and this quantifies the diversity of substrate utilization. You can also use Shannon Evenness, and this is going to measure how evenly distributed are the utilization by the community, right? So are they specializing, or they're just using everything that is available? And functional guild grouping just allows for higher level comparisons of different substrate utilization across the conditions, right? So together, these metrics provide a quantitative description of the community metabolic structure. And so, like we said, AWCD is often used as a measure of overall metabolic activity, and it provides a nice way to compare communities. You can even do this with different inoculum densities or signal intensities because they're going to get averaged. And researchers often use AWCD to track changes in microbial communities across treatments or time points. So this is like the overall metric to use, and it's like an index for you to determine how much microbial activity one community might have. And so you can compare different soil samples this way and sort of come to a conclusion that way. You can also use Shannon diversity, and Shannon diversity reflects the breadth of substrate utilization. So higher diversity means that a community can metabolize a wider variety of substrates, and this you can interpret it as metabolic versatility, right? So when the microbial community um, can can do more than just a few carbon sources, that means that it's a very versatile community. And this can explain how they can persist in the environment, right? And so again, you can use it to compare two different soil samples, and it is a very nice comparison. Now, Shannon evenness is the opposite. It describes how balanced the metabolic activity is across substrates. And so if activity is concentrated just in a few substrates, the evenness will be low, right? And so this helps you reveal resource use strategies, right? So for example, you can find niche differentiation this way. Some people call this the privatization of substrates. This is one way to really highlight the differences in resource use strategies, right? Um, Odin software makes it easy to do all this because these are all built in into the software, and it is easy to generate some graphs that are ready to publish here. Some more advanced analysis beyond these summary metrics can be done by analyzing EcoPlate data using multivariate techniques, right? Some common approaches are heat maps, where you just stack, different plates that you've read across different conditions, for example. You can do principal component analysis, cluster analysis, canonical correspondence analysis, and these are all sort of the more advanced technique where you would need more data, more plates, and more samples, right? And so if you want to learn more about these um, analysis tools, we have many resources available on our website. We have a webinar that explores the microbial community analysis tool, and we have a huge list of publication resources on our website that you can come and visit. Now, when you're presenting EcoPlate results, it is important to frame your findings in ecological terms. So rather than simply reporting stronger or weaker signals, we want to interpret what the carbon use pattern means, right? So for example, does the community prefer simple substrates or complex polymers? Are certain functional gills increasing or decreasing? These patterns helps us understand research use strategies and environmental responses, right? Um, so we have customers that come and they ask me, right, like, oh, so I have this data and I would like to interpret this. And so my very first response is always think about it in ecological terms, right? Are there functional shifts that you want to highlight? You got to think about the community and how it's interacting with its environment. And so some practical takeaways from this is hopefully you learn how to design a robust EcoPlate experiment, some recommendations for sample preparation, how to avoid some common mistakes, and how to confidently present EcoPlate results in your publications. Um, Another thing that is important is that EcoPlates are very complementary to all these other techniques like 16S sequencing. Hopefully you've seen some examples of this during this webinar. Lastly, I want to highlight Biolog's ecosystem of products and services, right? So Biolog is a microbiology company, and we have these three pillars that you can rely on. We have our instruments and software, where we have identification tools, phenotyping analysis tools, our Odin instrument, or anaerobic chambers, but we also provide consumables like media, um, PM plates, and we are even incorporating lab services where you can send us your samples for identification, for phenotyping, and we're fully certified and we're just here to help you make the best of your studies. Thank you very much. And with that, I'll take any questions right now, or if you have questions later, you can just email them to info@biolog.com. Thank you.